So thanks a lot again for this opportunity to present our work. So we're going to mainly talk how we apply machine learning in for understanding and engineering biological systems. So before I move to talking about machine learning, I want to remind you about very kind of one of the, let's say, a global pollution catastrophe which we are currently facing all over the world, and it's plastics. So plastics, you know, are very lightweight, versatile materials, literally used in almost all products in our daily life, and its production increasing annually. And since generally plastics were designed in such a way that they could be resistant to all sorts of environmental impacts, they are basically, they can stay within, so for instance, degradation of plastic bottles can take hundreds of years, and some plastics will stay with us for next millennia. So and our lab is very pragmatic, and we are oriented in a problem solving. We want to contribute somehow solving the plastic pollution by enabling basically biological plastic degradation using synthetic biology approaches. And these are just some other statistics. So basically, if we don't stop producing plastics with the pace what we currently do, in next 30 years will be more plastics in oceans than actually fish. And to give you another example, so this is a amount of the produced plastics. This is enough to cover this type of this area, and this is also enough to quite well cover this beautiful country. So you can imagine the amount of stuff we are currently producing. So and next I want to basically share our vision of how we tackle this plastic degradation problem and tools and technologies we're currently developing for designing microbial systems. So we started from the question how we can use the planet's basically biological power to degrade plastics. And the idea was simply to look for known plastic degrading enzymes in all environmental DNA. I'm not sure if everyone is familiar what metagenome is, but basically it's a DNA directly isolated from environmental samples. So microbes, you know that the microbes are everywhere and they have their own DNA. And what we can do that we can take a sample from environment, we can sequence them, and then using some computational approaches, we can then infer what kind of proteins there and what are the potential functions are these microbes are doing. And basically, very large scientific consortia sequence samples from all sorts of places around the globe, and this is we have this tons of this DNA data laying in public database. And to explore what was out there, we searched for literature for plastic degrading sequences, basically to identify any the sequences with the known plastic activity, and then they search all sorts of these metagenomic projects. And basically what you can see here is a map of various plastic degrading enzymes from environmental samples, and the dot represent identify plastic degrading candidates from nearly like 600 locations around the globe. And we identified around 30,000 plastic degrading enzymes in total with the potential to do this. What we also found that generally if you look at the pollution in those areas with identified plastic enzymes, you can actually find that there is some correlation with the plastic degrading enzyme diversity, both in oceans and actually in soil. And this work was quite well perceived in media, attracted a lot of public and scientific attention. Now our challenge is to test these identified sequences in the lab and actually to evaluate them of how good they are at degrading plastics. And to develop these plastic degrading enzymes, we need minimally to do two things. First, we need to basically take DNA, put to the cell and express it to sufficient amounts to active proteins. So remember, we only have a DNA, so the DNA needs to go through all sorts of biological processes until finally it can code. We can produce a protein that can do the job. So we are quite well studying the processes in between. And the next thing after you express the DNA, well, protein, what you need to do is also need to kind of engineer it. So usually it might be that the proteins are doing a slow job, or that they can't be applied in practice. So it would not be feasible to develop any process. And this is where we come with the protein engineering. We want to make a proteins basically more active and stable that can be further used for practical applications. So historically, we are a metabolism lab, and we were always looking at metabolic networks and how the enzymes regulate metabolic reaction fluxes, and etc. And one of the observations was always kept puzzling, that some enzymes, why were always some enzymes low expressed and some were like orders of magnitude higher. And we hypothesized that generally that DNA probably can potentially encode this expression levels and eventually the protein levels. And this is what was the first question when we started the lab at Chalmers. And I want to pay your attention at this figure. So basically it demonstrates mRNA expression levels of every protein coding gene across 17,000 RNA-seq experiments. These are experiments basically where you actually measure this molecule. And you can see, so you can see on average that some genes are low expressed and some genes are always high expressed, and they are kind of defined with a certain range. And the difference between low expressed gene and high expressed genes is basically 7 orders of magnitude. And these are so the same phenomenon observed in these. So for majority experiments, the basically expression of the gene lies just within a fold change. Fold change means just double. There is the differences between the genes. So from this gene to that gene is orders of magnitude. And these are virtually all experiments that were done in yeast. And the only common feature here is genomic DNA. So the question was actually how much of mRNA levels is encoded in the DNA. So I'm talking about looking at the DNA and telling how much of these guys are expressed from the DNA. And typically, DNA is a long sequence, and to study biological sequences, typically you want to use so-called convolutional neural networks with a combination some recurrent neural networks or some more sophisticated approaches that can capture long-range interactions in the DNA. And what you can get from this analysis is you can find the patterns that are responsible for biological processes, for instance, binding of certain proteins on the DNA molecule, and basically that then we can give some condensed quantitative interpretation of patterns that are encoded in the DNA as they relate to biological processes, for instance, such as gene expression. And indeed, just by looking at the DNA, we can predict quite well how much on average the gene is expressed. So here, what is shown is basically prediction just based on the DNA letters on the x-axis, and the y-axis is experimentally measured gene levels, and each dot is a particular gene. And the same phenomenon was also observed for other organisms, including human. So we can also what we learn, we can also interpret, and I will kind of demonstrate it with a simple example of, imagine if you have a cat classifier, you want to understand what type of, where AI or machine learning algorithm actually pays attention to which part. And in case of, for instance, cat, to classify a cat, it's mostly recognizing the face to determine whether this is a cat or not. And this is highlighted here. And the most simplest way to do that is you can take, for instance, a DNA sequence or a picture and occlude it with a black patch, and then compare actually the predictions based on your the occluded sequences versus the one which is not, and then you can find a difference. And if you do that for DNA, you can actually identify the regions that are mostly important, for instance, for gene expression prediction. If you find those significant peaks, you can take extract the letters that are behind those peaks, and then you can identify those motifs that are those patterns that are important in the DNA for predicting the DNA's the protein or gene expression. Sorry. And if you do that across all the DNA sequences, you can identify lots of so-called these motifs. And we also hypothesized that maybe some of the motifs would co-occur across all these regions. For instance, what I mean by co-occurrence, that if you could find some motifs in combination that are more frequently in combination than any of them on alone. And this type of technique is often, we borrow this technique from market research. So this is called market basket analysis, that typically analyzes the transactions of customers in the supermarkets. And from this example, what you can see that often when the guys are going to buy diapers, they always buy it with beer more frequently than on alone. And we also did that basically across all these regions. These are specific regions in the DNA, when we found that many of the rules, we term them rules, so co-occurring motifs, we term them rules, are co-occurring across the terminators, promoters, etc., in different parts. And this gave us a clue that there is interaction between the DNA sequence elements. And to demonstrate it further, you can see this is a transcription factor binding site, which is identified in promoter. And you can see, depending what kind of, with which kind of motifs it's co-occurring and where they are, you have quite a wide range of expression changes. So basically, that led us to the hypothesis that there is a strong interaction between generally, for instance, the promoters that are very important for transcription, terminators, or in many other parts of the DNA. But we made a simple experiment by taking a native promoters of yeast and switching to putting a strong terminator or weak terminator. Basically, we wanted to show that a strong promoter can become weak promoter depending on which terminator you put, and we've done that quite successfully. We also made a weak promoters by putting a strong terminator and boost the expression quite significantly compared to actually the native promoter. So generally, there is this kind of interaction across the entire gene structure, and this we identify with machine learning. So now I want to look at this picture. So the picture on the left is actually how AI makes faces, and the picture on the right is Picasso. And as a human, we have excellent engineering skills, but this is but generally this is how we make a DNA. So this is a beautiful art, but probably cell can distinguish what that these doesn't look like a real person and doesn't look like a natural DNA in that sense. So you might be familiar with these approaches, but I want to just to emphasize in general, there are kind of two main paradigms. There are so-called discriminative methods in statistical learning. There are discriminative methods that try to directly predict the outputs. They primarily used in supervised tasks. So for instance, the model gets its features like height and weight of these animals, and then you can distinguish whether it's a cat or a dog. And instead, the generative models trying to explain how the data was generated. Given a class, for instance, if I give a class, the one will tell what will be likely features like weight of height, for instance, of a dog. And one advantage of these generative algorithms is that you can use them to generate new data with the properties of existing data. And again, another example from synthetic biology, but I always like to show it with images. So using a combination of this generative and discriminative models, it's possible basically to navigate the faces space in a specific direction. For instance, you can make a person look younger or older, or you can change the sex or change age and etc. etc. So similarly, we apply the similar techniques, but instead of like changing properties of the face, we wanted to change properties of the DNA. So we trained the generator of the DNA of entire DNA sequence, including the promoter, like the coding region and terminator sequences, UTRs, and so on. And we use the predictor, which I shown that predicts nicely the DNA expression in such a way that you can use this predictor to navigate the generator to reach a specific value of gene expression. And this is can be demonstrated on this plot. So basically, all what these are shown is these dots represent a DNA specific DNA sequence, and the color represent kind of the expression, predicted level of expression. And you can see nicely that you can, by starting from very low, you can actually navigate the sequence to specific gene expression level. And this is nice because typically, in order to, if you want to make a strong promoter, you typically put it, you create millions of DNA variants and do very kind of tedious screening experiments. And this is, but instead we wanted to do that in the computer without actually relying on the directed evolution experiments that you do in the lab. So, and if we compare the generated sequences to the natural ones, we can see actually that they resemble properties, the expected properties that exist in nature. And when we sample the sequences, then we actually synthesize them, put to the yeast, and we got some sequences that are very far from what is known in yeast biology, but they were even more expressed than the highest expressed genes in yeast. So I guess the most important message here that we don't need to do these screening experiments with millions of synthetic sequences. We can, well, just learn everything from natural genomes, and this can be, and the nice thing here that this virtually can be applied to any organism, or potentially in a specific tissue if you want to have a specific expression in a specific tissue, and where you or something unknown where you don't know anything about gene expression regulation. You just need to have some data from those organisms. Well, summarizing this part, I want to remember three things. So first, the DNA encodes for gene expression levels with over 80% of mRNA level of variation, which is encoded in the DNA just from the DNA sequence. The second, generally the deep neural networks from kind of classical statistical point of view are often viewed as black boxes, but however, it is possibly quantitatively interpret them to extract the new biological knowledge. For instance, we can quantitatively link effects of gene expression at the nucleotide level resolution. And the third, generally these machine learning techniques, generative machine learning techniques, allows actually learning directly from genomic data in any species, no need for synthetic libraries and any tedious screening. So this is just some publication describing with more detail. So, right. So coming back to our initial problem, so we can express, let's say we can express proteins. Now we want to, what we need to do is actually to make them, more effective. And how can we do that? Generally, it's quite challenging to do this. So traditional. I will explain how it's done traditionally. So protein engineering begins by you have your molecule of interest. You basically introduce random mutations, then you do some selection, and then you take the improved variant again, introduce some mutation, diversify, basically by introducing mutations, and do that in many cycles. And just a few years back, a Nobel Prize in chemistry was given for this technique, and maybe many of you saw a great talk from Francis Arnold here at Chalmers, and this is also often depicted as climbing this mountain of complex landscape. The problem, however, in the reality, these protein landscapes are looking like more like this. So instead of climbing the landscape, you end up in screening this dead protein sometimes by hitting these islands of activity. And what would be great is actually to have that we would only be able to navigate the functional sequence space and not just random sequences. This is just another example that showing that if you introduce normally several random mutations to protein to get something that is active is very unlikely, and this is some statistics so that you understand what the complexity of the proteins are. So, you know, because of this huge space of amino acids, you know, a very short protein of 100 amino acids can be done in these many ways. So here is another example how you know, AI can do the images. And as I said, that these people don't exist in nature. These are all just a kind of a sample of phase from phase space, random sample from space. And basically we wonder if current machine learning technology is ready to generate proteins, same as AI learns this relationship between millions of pixels to generate faces. Can we actually use this technology to learn physicochemical relationship of amino acids in natural proteins to be able to generate new sequences? And we challenge this. So basically, to make this very complex biochemically protein such as Malate dehydrogenase, which is a long protein, it's active only in multimeric form. It has 2 active sites and quite the diverse family of sequences. And this was just a reaction that we can quite easily monitor in the lab. So for this, we trained a so-called generative adversarial network. I'm not sure if some of you might be familiar, but the core idea of the GAN is based on the indirect training through the discriminator. So, which is itself also updated dynamically. So basically means that the generator is not trained typically to minimize the distance to a specific training example, but rather it does so by fooling the discriminator. And this enables to learn the model to learn in a supervised manner. So you train two networks iteratively. For the generator, you try to map a random, well, here it's so-called noise vector or latent space to the sequence distribution, such that given a random input vector of numbers, the generator produces a protein sequences, which is then scored by the discriminator network compared to actually to the natural protein sequences. And the generator tries to fool the discriminator by generating the sequences that looks like real ones, and you continue until you get the desirable effect. And then you can remove the discriminator, take the generator, put the random numbers, and basically generate the sequences and test for various properties. And of course, the challenge is like when you generate in the images like this one, it's easy to see that wow, it looks like a nice person. But with proteins, it's harder to do so because, as I mentioned, just single mutation can often likely kill the protein, and it is nearly impossible to detect. So, so we started simply by like doing the training with many steps and seeing how the generated sequences look like, similar to the natural ones, and develop all sorts of monitoring matrices to generate this. To monitor the progress of learning, and after about 1 week of training, you can actually get proteins that looks like natural ones. So this is a so-called multiple sequence line where you stack many sequences and you can compare them. And these lines represent the variation in the blue one. The orange one is in natural sequences, and the blue one is generated. And you can see that actually the generated quite closely resembles the natural sequences, and these are all these regions highlighted here represents the highly conserved regions, which are actually corresponds to the active site of the protein, and we could quite well capture that with some tricks, technical tricks. I don't know if you kind of if someone is interested from technical perspective. So what was interesting to observe that the choice of loss function was important for sequence generation. We tested many loss functions, and basically you can see this is a training progress, and this is on the y-axis so-called Blossom score, so which basically signifies to some extent the likelihood that amino acids is not appearing by chance, and that was quite for us important that we could not get a proper sequence training for a long time to play around with these losses and architectures and so on, etc. Anyway, we express this generated sequences Nicolai, purify them, and verify their activity. This is example some of them. So this is just a reaction monitoring progress. This is actual natural controls, and all of those with the blue ones are actually the generated sequences. And to demonstrate this further, you can see this histogram, right? And it shows how similar the synthetic sequences are to the closest enzyme in nature. So these are mutation numbers. And generally creating diversity such as 100 mutation or even few dozens using experimental directed evolution approaches is very challenging, if not sometimes possible. So remember, I showed this figure basically in the beginning, that by introducing even 8 mutation, there is a little chance to have anything active. This is just another example that we demonstrated that they are doing specific reaction. This is a mass spec data, and we can also show that some of them are even doing something more than natural ones, so a little bit more active. And the cool thing was that actually that some of the enzymes that were more thermostable than natural one. This is I can't explain yet biologically, but it was an amazing result to observe that it's something, learn something that makes it super stable even in a very high temperatures. So I guess just to cut story short, making functional protein diversity is very important if you want to change protein properties. And we believe that it can be facilitated by machine learning algorithm. So the best thing about generative models that allows this learning of physical and functional protein concept automatically in unsupervised fashion without a need of expensive labeled data. And we basically created the first functional protein entirely using AI approaches, and now we can kind of use this technology for practical applications. Also very happy to see that these approaches that we develop are now official part of the deep learning Coursera curriculum, which is the largest machine learning course on the internet with tens of thousands of participants. So, and now I'm going to be a little bit kind of first time presenting this part. This is ongoing work, but I also want to show other areas where we do the machine learning applications. So probably it might be that many of you familiar with so-called a concept of neural style transfer, which is basically while using an image domain. So it's a class of machine learning algorithm that manipulate digital images in order to adopt appearance or visual style of one image to another one. So in this case, you can see the example of Mona Lisa and the sky under the stairs from Van Gogh, and if you combine them, you can have a Mona Lisa in style of Van Gogh. Well, this was simple example. This is more complex example, and where the person just talks, right? And you can actually make it in style of Obama, and you can see that. I mean, you can imagine how many good and bad application of this technology, so-called deepfake technology, but it nevertheless shows the point what you can do with that. And what we particularly were interested in is how to make proteins more stable. So, for instance, stable proteins can stay active much longer under different temperature conditions, and that's very important for industrial and biomedical implication. And particularly, we wanted to make normally active proteins that are active in mesophilic ranges. So mesophiles are like about active around 30 degrees, and we wanted to make them in more thermophilic conditions with let's say if they are active in high temperatures. And the challenge is that you normally need to have lots of paired label data if you want to kind of to let's say to have an enzyme from certain family class. You need to have lots of data in order to kind of infer something about that enzyme if you want to for specific enzyme family. And in reality, you don't have this paired example. So for instance, if you want to see in a classical image domain, if you want to see, let's say, some picture of some painting of Van Gogh, who was painting that 250 years ago, 200 years ago, forgot when he lived. But if you want to look what he was seeing and generate from his picture a photograph, that would kind of require you to have normally a paired example. But actually, if you do some sophisticated machine learning approaches, you just can learn general features of Van Gogh and then translate the pictures between between the two different domains without having paired examples. The same goes with the biological sequences. You normally don't have any paired examples. So in essence, what we did this again. This is based on GANs with more sophisticated training. So you take a mesophilic sequences and you do some mutations, so you produce a thermophile sequences. So the plot on the left depicts actually the temperature shift of these in-style sequences, as opposed to randomly mutating the sequence. And you can see that we can actually have a quite significant temperature shift from the sequence. This is in silico data, but we using just purely based on amino acid sequences, we can actually predict the temperature shift quite well, and thus monitor also the results and network performance. And on the right, you can see that these blue navy colors represent the thermophiles, the cyan colors represent mesophiles, and the yellow one, the generated thermophiles, so they are closer to actually the natural one, the thermophiles, and also this kind of brownish, or they're closer to natural mesophiles. So we can kind of see that actually the network is doing something to sequence, and that actually makes sense. And now we want to kind of, before we do the lab experiments, we want to relate the sequence stability, or basically to do something still in the computer before we can actually do lab experiments. And what we do here is so-called molecular dynamic simulation. Basically, the idea of all this molecular dynamic simulation that you put a full atomic protein molecule with thousands of atoms in the box, filled with water under the constant pressure and volume and specified temperature. And then what you do, you monitor behavior, all these atomic jigglings at the picosecond scale. And what is shown actually here is a simulation for 100 nanoseconds. On the y-axis is this what I showed, these atomic jigglings that are happening with the protein in this box, and these are different temperatures, and these are different models. But you can see that normally what you want to have is this line that would be below this wild type sequence. This is a control sequence. And you can see that in this case, we can actually succeeded to do one below. And I guess the challenge here, as I mentioned, is how to monitor this any kind of key performance, the KPIs. It's hard to know where to stop the training of the model. And what is shown here is actually just different variants of the model. And you can see normally that this is a substitution matrix that from the wild type and optimized in each of the different models during the basically during the trainings. You can see that there is some kind of consensus, but generally they at least quite variable, and typically each mutation kill, as I said, killed the protein. Nevertheless, what's nice about this method that we can actually constrain the number of mutations and preserve some residues that we think are important. And in this case, the proteins that had actually active site preserved, where it seems to be are well, let's say more robust in this sense, and also kind of indicating that they might be potentially more stable. So take-home message that style transfer has a potential to be applied for protein optimization. We of course still are doing experiments. This is ongoing projects. So another question we were kind of interested in, and this is how does the actually protein levels arise from the sequence. So to oversimplify the protein problem, well, let's say protein abundance problem, the protein is synthesized using this process, so-called transcription translation. This is a protein synthesis part. and then it is also degraded. But if you have like a tight balance between the two processes, you normally what you observe in the cell is this is some level of the protein. And if you look at them, generally this degradation rate of the proteins, typically they differ between the different proteins over orders of magnitude. So we hypothesize that actually the protein sequence can have information about the protein abundance in the cell. And we ask the question how if we can predict protein abundance from its amino acid sequence alone. And we kind of a little bit inspired by natural language processing topic. We wanted kind of to investigate what type of a gram of amino acid combination dictating this protein abundance. And to do that, well, it's a bit oversimplified slide, but generally we train a so-called BERT transformer model to predict from amino acid sequence the protein abundance. And well, it's not the actual BERT, but it's generally to see that it's a very like deep neural networks with hundreds of millions of parameters learned through training. And if you do this exercise, you can actually have a model. Well, it seems to be that amino acid sequence is informative about protein abundance. So this is only based purely on sequence data on amino acid ladders. You can quite well predict the variability of protein expression in the cell, and each of these represent just different protein. What's nice about BERTs that they have this so-called self-attention mechanism. Is the way how it's an intrinsic way how model, let's say, learns what model pays attention in the sequence in order to make it predictions. And you can get this so-called self-attention matrices, squeeze them, summarize, and you can have per each basically amino acid residue, you can amino acid sequence, you can have this so-called attention profile, and you can correlate this attention profile with all sorts of biological properties, and you can see that generally you can find that even though it was never trained on nothing except of abundance, you can find something that is actually relate to protein conformation, stability, polarity, etc. But what was also interesting to find that actually that many of the things are related to cost. So for instance, the more highly abundant proteins, it seems to be cheaper to do it if you calculate the cost that is associated with every single amino acid synthesis. And what we next wanted to do, we wanted kind of to exploit this model to basically tweak deterministically the sequence in order kind of to change the in this case abundance. And we came up with this PhD student Philip, who will show the picture next, came up with this very nice approach that can actually possibly to do that. So when you have what basically BERT encoding does is it produces so-called embedded space of it's a long vector of 1024 dimension for each of the residue of amino acid, and basically you can represent it as a numerical point clouds, and the trajectory where these points are laying is associated with with abundance. So in principle, if we can shift this each point along this trajectory, we can actually replace these positions and make the proteins more highly abundant. The trick is how to do that in 1024 dimensions. It's quite unclear. And what Philip came up is actually that you can use somehow then I had a very nice picture of him. Well, anyway, you can actually do proxy trajectory that basically that would map this 1024 dimensional space to one dimension, and then you can actually move within one dimension, and they are kind of, the nice thing about them that they are ranked in a way, they're ordered in such a way that correspond in abundance, and you can show that here. So this is a unidimensional projection of those points on the UMAP space, and this is a actual prediction. So they in essence are BERT orders them in such a way that they are predictive of your target function. And I can just show you how practically that here he's very fantastically frustrated probably. So basically we wanted to have this sequence Malate to change to something that is more highly expressed. And the way how you can kind of do it, you start by navigating to some number, and basically well some random jump in the unidimensional space, and then just in order to convert this space to amino acids, you just look at the nearest neighbor on that projection. For instance, in this case, that was letter O. Then you can actually change your original sequence, and actually, if you change in this way your original sequence from this guide borrowed from this guide sequence, you can actually increase the prediction. And if you do this exercise for our proteins, you can actually, on average, predict your mutation. So the prediction of protein abundance increases on average by double, but just roughly mutating 10 residues. And this would be highly unlikely to achieve by chance, because and generally explore with any kind of optimization technique, because the space is quite enormous. So this is a nice general technique to how to navigate a relevant manifold to the target function. And so if you look at those which amino acids get substituted, these were mostly, we found that in principle the polarity of the protein changed, and that's kind of led us to the part that we actually think that it could be also the more abundant, it could affect the stability of the proteins. And we indeed investigated that again with molecular dynamics simulations. So what is shown here on the x-axis is a proteins, carbons, backbone, atomic position. These are several different proteins, and if you do molecular dynamics on them, you can see these two lines. The green one is a wild type, and the blue one is in your mutant, and you can see that it's always below this line. So this indicates that generally that the jigglings in those atoms are kind of more, or those proteins are more constrained, so they are kind of less, so they are more robust and such in this way. Yeah, I want to show you nice. As always, it never works, but it's supposed to be a nice. Does it work? Please. Yes. Okay. So what you see here is this. The mutant is basically this blue one, and the green one is the wild type molecule protein. Sorry, and you can see that it's more kind of jiggling. The blue one is more robust. Then we did an experiment for this particular protein, and you can actually show that generally, if you compare the exponential stationary phase in the wild type and our mutant, you can see that more protein remains in our mutant in a let's say stationary phase compared to the exponential in yeast. So in summary, I showed you what we can do with how can we use these generative models to design the functional proteins. However, there is still quite a few challenges, which you kind of basically outline here. It's generally hard to look at the proteins. There is no metrics, and we are actually working kind of to decide how can we, to know that sequence is likely is active or not active. So in this example, for instance, is again our Malate dehydrogenase. So if you see that this is a superimposed with the homology structures of the cyan ones are active enzyme, and the blue one is an active enzyme. What you can see, this red color is actually shows the position where amino acids were different between active and inactive. So if you imagine that we are learning only on the monomeric proteins, right? So on the monomeric part, it's somehow still we need to make sure that somehow these generative models kind of also let's say try to understand what is the like a let's say multimetric form, and this is quite unclear. We're still working, and this is where we believe that the molecular dynamic simulation would help. In general, the solubility is quite an unsolved issue generally for any kind of recombinant protein production. So we get lots of, when we test them, lots of them are unsolvable. So concluding, what I want to summarize, our approach in what we do. So as I show you, we are heavily relying on machine learning technologies that we combine both the systems and synthetic biology frameworks, and we follow this philosophy based on famous Richard Feynman's quote: "What I cannot create, I do not understand." Specifically, on machine learning, I envision that we will be able to learn the patterns from natural systems and to understand how evolution encoded quantitative information in biological sequences. And I believe that we will be able to put a predictive number to all the processes in central dogma towards predictive dynamic models of gene expression and physiology on the entire systems level. And we create and verify the findings experimentally in synthetic biology settings, basically to create new molecules and proteins such as DNA proteins for practical human applications. And if our predictions based on derived knowledge and biomolecule design work experimentally, we can then kind of start saying that we understand the system. And I would like to thank all of you also for listening, and also by our division, and specifically the lab members whose work I showed, all our collaborator, and of course funding sources. Thanks a lot, and I'm very happy to take any questions you have.
Thank you. Very nice talk. I will stop the recording and then we can jump onto the questions.